Exploring Life. Chapter 17, Evidence of Evolution. Part 1. Natural selection is the process that explains how species evolve and adapt over time. It's one of the most important ideas in all of biology. This concept was first introduced in the mid-1800s by Charles Darwin and Alfred Russel Wallace, two naturalists working independently but arriving at similar conclusions. While both contributed to the idea, it was Darwin's version, supported by years of detailed observation and research, that laid the foundation for what we now call modern evolutionary biology. This is Charles Darwin in 1854, just 5 years before the publication of On the Origin of Species. The image beside him is the only illustration included in the book. It shows how species evolve and branch out from common ancestors over time, a simple but powerful representation of life's diversity unfolding through natural selection. Darwin's natural selection is based on 4 key principles. First, variation. Individuals within a population differ in their traits. Second, competition. Because resources like food and space are limited, individuals must compete to survive. Third, differential reproduction. Those with traits that offer an advantage are more likely to survive and pass those traits on to the next generation. And finally, adaptation. Over time, beneficial traits become more common in the population, helping it adapt to its environment. This table connects the core principles of natural selection, as described by Darwin, to how they are interpreted through modern biology. Darwin spoke of variation among individuals. Today, we understand that variation arises from mutations and is reshuffled through sexual reproduction. He described a struggle for existence. We now model this as selective pressure, using tools like the Hardy-Weinberg equilibrium to predict how traits persist or shift in populations. When Darwin observed that individuals with certain traits were more likely to leave offspring, and that these traits became more common over time, he was describing what we now recognize as changes in allele frequencies, a process we study mathematically in population genetics. Darwin lacked the tools to explain the how. Concepts like population genetics and the Hardy-Weinberg model would come more than 50 years later, but he accurately captured the pattern. How did he manage it? Charles Darwin wasn't a trained biologist. He studied theology, didn't have a steady career path, and joined the HMS Beagle mainly as a travel companion and amateur naturalist. But what Darwin did have was an exceptional gift. He was deeply observant, and he wasn't blinded by the mainstream beliefs of his time. While others accepted that species were fixed and unchanging, Darwin paid attention to the small differences. He noticed how species varied from island to island, and how those differences seemed tied to their environment. These quiet observations led him to a bold idea, that species change over time, and that natural selection is the force guiding that change. When he published On the Origin of Species in 1859, it sparked both praise and backlash. Many celebrated its scientific depth, while others rejected it, especially those with strong religious or traditional views. But Darwin's theory stood on evidence, not opinion. And over time, it reshaped biology, offering a natural, testable explanation for life's incredible diversity. Today, it remains a foundation of modern evolutionary science. Today, the theory of evolution is supported by overwhelming evidence from every corner of science: genetics, paleontology, embryology, comparative anatomy, and more. It's not a guess or a belief, it's a well-tested scientific explanation. The evidence is so extensive, it's often compared to the case against a flat Earth. Just as we can confidently say the Earth is round based on centuries of data, we can just as confidently say that evolution is real, ongoing, and explains the diversity of life around us. Many of these lines of evidence you've already encountered in previous lectures. In this section, we'll focus only on the most compelling examples that we haven't covered yet, the ones that help complete the bigger picture. Fossil records provide a powerful historical narrative of life on Earth. They let us look back in time and see how organisms have changed and evolved over millions of years. And they're not far away in some abstract sense, fossils are all around us. We're walking on top of them every day. They're part of the very ground beneath our feet, woven into the layers of Earth's surface. Some of the most important fossils are transitional forms, species that show traits of both ancestral and more modern groups. These forms help us trace the steps of evolution and understand how major changes happened gradually, over time. Together, fossils create a timeline that connects ancient life to the species we see today, showing us just how deeply all living things are connected through Earth's evolutionary history. These are transitional forms, snapshots of evolution in progress. On the left is Archaeops, an early amphibian that still carries traits of its fish ancestors. You can see it in the flat skull and the limb structure, adapted for both life in water and movement on land. On the right is Archaeopteryx, a remarkable fossil with the skeleton of a small dinosaur but covered in feathers like a bird. It's one of the clearest links we have between reptiles and modern birds. We'll come back to these and other transitional species later in the course, when we explore the evolutionary journey of vertebrates in more depth. Some species alive today look almost identical to their ancient ancestors in the fossil record. We study a fossil for years, until it turns up alive and well. That's exactly what happened with the coelacanth, a deep-sea fish thought to have disappeared with the dinosaurs over 65 million years ago. For decades, it was known only from fossils, until a living specimen was pulled from the waters off South Africa in 1938. This fish, with limb-like fins, offers rare insight into the evolutionary transition from water to land. On the left is Ginkgo biloba, a plant that has existed for over 200 million years. Its fan-shaped leaves are unlike anything else, not quite pine needles, not quite flowering plant leaves. Ask any botanist, it's a true evolutionary in-between. For a long time, ginkgo was believed extinct in the wild, until a small natural population was rediscovered in eastern China around 1916. Today, it's planted in cities around the world. And if you've ever caught a whiff of something that smells like vomit in the fall, you're probably stepping on evolution. That's the fruit of the ginkgo tree. On the right are horseshoe crabs, virtually unchanged for 450 million years. Despite their ancient lineage, they're in high demand. Their blue blood contains a compound that detects bacterial toxins and is essential for testing the safety of vaccines and surgical tools. Evolution may have taken it easy on them, but modern medicine sure didn't. Since once acquired mutations are preserved, our genomes serve as a historical record of evolution. Comparative genomics examines the genetic code of different organisms to uncover that past, revealing relationships and timelines that fossils, bones, or behavior alone can't fully explain. It's a relatively young field. While some discoveries have already been made, many more are still waiting to happen. You'll actually get to dig into genome-based history yourself in one of the labs for this chapter. Comparative anatomy helps us understand how species evolve in response to their environments, sometimes in similar ways, sometimes in very different ones. Homologous structures are features that may differ in function but share a common evolutionary origin. They reflect shared ancestry, even if natural selection has shaped them for different purposes. In contrast, analogous structures perform similar functions but evolved independently. These arise through convergent evolution, where similar environmental pressures lead to similar adaptations in unrelated lineages. This color-coded diagram highlights homologous structures, specifically, the same pattern of bones found in the human arm, a lion's paw, a bird's wing, a bat's wing, and a whale's fin. But it doesn't stop at limbs, or just these examples. Similar structural patterns appear across many body systems, and in a wide variety of animals. Homology reaches far beyond just a few famous examples. It reflects our deep evolutionary connections. Analogy is when the same challenge gets a different solution. This figure shows two different types of wings, a bird wing and a butterfly wing. The bird wing is supported by bones and covered in feathers. The butterfly wing is made of chitin and covered in scales. These structures clearly evolved independently in response to the same need, flight. More broadly, we see convergence everywhere in nature. Marine animals like fish and whales share similar coloration, lighter undersides and darker backs. Hibernation appears in both insects and mammals. Complex social behaviors evolved in bees and elephants. Even warm-bloodedness, maintaining a stable internal temperature, arose separately in birds and mammals. Vestigial organs are structures that once had important functions in ancestral species but have since lost their original purpose. Their presence is strong evidence for evolutionary change over time. They're like biological footprints, showing how species have adapted and shifted from their past forms. The human appendix is a small, tube-like organ attached to the large intestine. In our herbivorous ancestors, it was part of a larger cecum used to help digest plant material, specifically cellulose. Some snakes, like pythons and boas, still have tiny vestigial hind limbs, small bone structures buried in their bodies. These are remnants from ancestors that once had fully developed legs used for walking. These examples show how evolution doesn't always erase structures entirely. It sometimes leaves behind traces of the past. This image highlights the nictitating membrane, a translucent third eyelid found in many animals, like dogs. It can extend across the entire eye, helping to protect and moisten it while still allowing some visibility, even when closed. In humans, this structure is reduced to a small, non-functional remnant in the inner corner of the eye. It no longer serves its original purpose, but it remains as a vestigial trace of our shared evolutionary past with other mammals. Embryology is the study of how organisms develop from fertilized eggs into fully formed embryos. When comparing species like fish, reptiles, birds, and mammals, we see remarkable similarities during early development, such as the presence of tails or pharyngeal arches, even in animals that don't retain them as adults. These shared developmental features suggest a common ancestry, supporting the idea that today's diversity traces back to shared evolutionary origins. This image shows a human embryo at about 5 weeks of development. At this stage, all vertebrate embryos, including humans, share key features, such as a visible tail and pharyngeal arches. In fish, these arches develop into gills. In humans and other land vertebrates, they evolve into parts of the face, jaw, and neck. The striking similarities in early development offer strong evidence of common ancestry among vertebrates, even if the adult forms look very different. In the second part of the lecture, we'll explore artificial selection, how humans breed organisms for desired traits, and how it mirrors natural evolution. Then, we'll dive into the vast timeline of life on Earth, covering major events like the Cambrian explosion, mass extinctions, and shifting continents that shaped evolution. Now it's time to pause and check your understanding. This isn't a test, it's a chance to reflect. Take a moment to think about which ideas felt clear, and which ones you might want to revisit. Understanding evolution is about seeing the big picture and the processes that connect it all.